My name is Professor Karen Olsson-Francis. As Maria Pass mentioned, I'm a professor in geomicrobiology at the Open University. And the group that I lead is very much looking at not just the scientific, but also the governance and ethical challenges associated with astrobiology, which is something that we generally don't think about, but it's very much an issue when we think about space exploration. In this presentation today, I'll very much focus on some of the scientific work that we do, and particularly looking at the application of field work, so analog work, and applying that to laboratory simulation experiments to understand not just habitability, but also biosignature formation, so molecules which can be used to look at evidence of life elsewhere in the solar system. So, as a microbiologist, I'm going to start by introducing you to the concept of microbial life and extremophiles, why they're important to astrobiology as a discipline, before going on to discuss the role of both analogues and lab-based simulation experiments in searching for life elsewhere. Ooh. So, as a microbiologist, when I think about life elsewhere in the solar system, I predominantly think about single-cell organisms, so microorganisms. And we think about them on Earth, we know that they're basically everywhere. This image here is taken from a swab of a mobile phone. So just be prepared when you think about microbes, they are literally everywhere. And there's approximately 5 million trillion microbial cells currently existing on Earth. And they are the largest source of organic carbon. And they do so many roles on Earth. You know, they shape the environment they live in. They're very important for our health and well-being of humans. And I think more importantly, when we think about life on Earth, some of the most, earliest evidence of life is microbial life. And the record suggests that life existed on Earth about 3.5 billion years ago. And for life to exist, what do we know? Well, based on Earth, we know that it has 3 key requirements. Firstly, it needs liquid water, so a medium for it to grow in. It needs bioessential elements and an energy source. And when we think about bioessential elements, we predominantly think about CNOPs, so that's carbon, nitrogen, oxygen, phosphorus, and sulfur. And these are the main components of building blocks of cells. And then finally, we also need the energy source. This is something that drives biological processes within the cell to live and survive. And when we think about that in humans, how we obtain energy, we get that from the food that we eat and the oxygen that we breathe. But microbial life is a lot more adapt and a lot more evolved than we are, as it is evolved to able to use a wide range of chemicals for energy. So here's an example of this. When we look at organisms in general, there's 2 dominant ways in which they can do it, either by chemicals and light, depending on the environment they live. So for example, on the surface of Earth, if we look at plants, for example, they can use energy from the sun, so they're phototrophic. And for organisms such as plants that use oxygen, they can use photosynthesis to produce energy. Whereas on the other side, we can use like chemicals. So humans are an example of using chemical energy to produce energy. So humans are chemoheterotrophs, so they can utilize energy from food and respire oxygen. But I think from an astrobiology perspective, when we think about energy sources, what's most interesting are these group of organisms called chemoautotrophs. Now, these are organisms that can contain energy from inorganic sources. So, for example, they could produce energy from hydrogen, sulfur, iron, nitrogen, etc. And they're able to use carbon from carbon dioxide as an organic carbon source to produce organic carbon. So in subsurface environments, when we're looking at these environments, the chemoautotrophs very much dominate primary production. So they're very much the driving force behind any microbial life within these environments. So as well as having key requirements such as water, bioessential elements, and energy, the environmental parameters in which microbial life lives is also really important. If we look at this figure here on the right, familiar life, this is where we as humans live. It's very much defined by the pH and temperature in which we can live. So temperatures range between, about 0 to 40 degrees, and about, the water we drink about, from about 4.5 to about 8. This is kind of how we define as life where humans can exist. But microbes can push this envelope of life. They can push these limits, the temperatures that we can grow, the pHs that they can grow. So if we think of an example of this, for example, Yellowstone hot springs, we know that we can have, really acidic and really hot environments, and we know that microbes that can live and survive within these environments. And we can know that microbes, in contrast to these high temperatures, can live in really cold environments as well. So, for example, in sea brine. And an example of some of these videos here, these microbes, these extremophilic microbes, can live in some really interesting places on Earth, everything from hot springs to hydrothermic systems. So this is Pyrococcus furius, which has an optimum growth temperature, about 100 degrees. And we have some microbes that can live in really cold environments, such as the Psychobacter fragmatius, which can live at -80. And then we have this really interesting group of microbes that can live at really multiple extremes, such as desiccation and UV radiation. And this helps them to survive in environments such as deserts, where they're exposed to high levels of radiation, dry environments as well. And then we have organisms that can survive a multitude of extremes. So a prime example of that is an organism called Deinococcus, which can survive high levels of radiation, low temperatures, limited water, etc. And studying these polyextremophiles is really important when we're looking at defining and pushing the envelope of life. So these polyextremophiles, so microorganisms that can live in more than one extreme, really enabled us to understand the limits in which they can provide. So we can delineate the envelope of habitability by looking at these conditions and kind of understanding whether they, the limits which life can exist. So we can apply what we know for microbes on Earth to looking at potential life and habitability elsewhere in the solar system. So, for example, we can look at this image that was recently published, where they've taken the envelope of life, so looking at salinity, pressure, pH and temperature, and compared that to other celestial bodies within the solar system. So the top left graph here demonstrates what we know about microbial life from culturing and from environmental studies. And then we can apply this to what we know about some of the other bodies, which we know that could exist within the solar systems. So the same laws of chemistry and physics apply these principles, so we can extrapolate these to start to look at habitability elsewhere. Now, you can look here if we can find these sweet spots where the envelope on the left, the top left, where life can exist on Earth, could actually be feasible on some of these other celestial bodies. This figure obviously doesn't look at all the other parameters which are really important, such as UV radiation and desiccation, but it gives you an understanding where potentially the conditions in which life exists on Earth could potentially exist elsewhere in the solar system, which suggests that potentially habitable environments may exist. So let's look at Mars in more detail. Mars is a prime candidate for looking at habitability. Now, although the surface of present-day Mars is really cold and dry and highly oxidized and exposed to UV radiation, ionization radiation, and are considered inhospitable to life, about 3.6 billion years ago, where life was evolving and existing on early Earth, the conditions could have been more conducive to life. Large fluvial systems could have existed with a possibility of warmer climates and a denser atmosphere that could have provided the protection for the UV and cosmic radiation. But as the atmosphere was lost, Mars became more arid and dry and highly oxidizing, which now means that the conditions on the surface of Mars are deemed inhospitable for life. So if we were to think about life now on Mars, extinct life, could be evidence of life, could be within the surface of Mars, but if we're looking for living life, potential environments which habitability could exist on Mars, we're looking very much more in the subsurface environment. And we have extensive information from the surface of Mars, from both rovers and orbiter data, which suggests that the conditions on Mars may have once have been conducive to habitability. So, for example, data from MSL indicates that at Gale Crater, there could have been a large fluvial-lacustrine environment that was subject to physical chemical variations, such as redox conditions and evaporation, which could have initially have allowed life to grow and form over time. So, why do we think this? There was a pivotal paper looking at Yellowknife Bay in Gale Crater a few years ago, which suggested that the key requirements for life could have existed on early Mars. So if we go back to that initial diagram that I showed you at the beginning, where the three requirements for life are liquid water, bioessential elements, and also an energy source, the information from the geology of the site suggests that liquid water was available. There were the key elements: carbon, hydrogen, nitrogen, phosphorus, sulfur, and oxygen could have been present, and also a redox environment which could have been able to support those chemolithotrophic, those microbes that can utilize inorganics as a chemical, as an energy source, and also building on that. Once those cells dies, they could have been utilizing, microbes could have been using the dead cells as organics to grow. So they're potentially looking at this information about early Mars. It is possible that it could have been putatively habitable within that environment. So to study habitability within this environment, it's very expensive and very difficult to get to Mars. We have a lot of missions planned, and then really key important missions with Mars Sample Return, ExoMars, all planned for the future. And we do have a lot of information already about the Mars environment. So to really understand that and look at habitability and how we can get the most out of these missions, it's really important to do work on Earth. Now, as an astrobiologist, that can look like anything. It could be working in extreme analog sites, so doing field work. Here's a picture of me collecting samples many moons ago from a salt pool in Ethiopia. It could have involved using lab simulation experiments. So that's using equipment that has been developed so we can start changing different parameters and conditions to start looking at potential life. It could involve using thermochemical modeling. So this is where we can take direct information from missions. So for example, the composition of rocks, and model the brine composition, so the water environment, which potentially could have been habitable. So what we do in my group is kind of a combination of all these things. It's combining field work, lab simulation, and modeling to start to study and look at habitability, and if, for example, early Mars was inhabitable. So, what is an analog site? Well, an analog site, we talk about this in astrobiology quite a lot. These are actually sites that on Earth are assumed to have been either now or still similar to another planetary body. So the biological conditions could be similar, the geology could be similar, and we can use information from studying these analog sites to extrapolate to other planetary bodies. And this is really important for understanding and interpreting and validating data received from orbiters and missions or rovers as well. Now, there's when you think about analog sites, it's not one analog fits all. It's very much specific to not just the planetary body, but the time and the geological time that you're looking at. So, for example, if you wanted to study, for example, the Neochian-Hesperian transition during Mars, the history of Mars, you could maybe look at an arid, dry environment on Earth. Here in this image here on the left, on my left, you have an image of Atacama Desert, where there's a lot of astrobiology researchers looking at microbial survival and potential metabolisms within these environments, and then applying that to right, which is the Martian environment. So very much, they look very similar. when you look at these images, but it's really important to apply the information that we learn about these environments on Earth to look at these other planetary bodies. So, analog sites are probably, there's so many analog sites out there that, I'm not going to list them. Here are some examples of the analog sites that my group's been in the last 2 to 3 years. So everywhere from looking at the salt pans in Botswana, right through as an analog for Mars, right through to Svalbard, using the glaciers and there to look at processes on icy moons. So it is very much dependent on the environment, depends on what you're simulating and what kind of work that you're interested in doing. For us, most of the work that we have been doing has been work in the global south, so a lot in Africa and in South America. And for us, it's really important that we do this research in an ethical way. So making sure that we work with local communities and we work with local universities. So again, an example in this bottom image here is us in the field in Botswana with colleagues from the University of Buist in Botswana. And the relationship we have with them, it's not just they're our tour guides, we work with them, we work in their labs, we share analysis, we interpret data together. So it's a true collaboration. And I think that's something, where we as astrobiologists have to be really careful to make sure that we continue to do this, because these are, really novel sites. So it's just making sure that we have that good working relationship when we do some of these analog work going forward. So I'm just going to look at one of these sites in more detail. We will look at Axel Hyberg in the Canadian High Arctic. Now, this is an environment which is really difficult to get to. It involves helicopters, and it's not the most easiest analog site to get to. But it's really interesting from an analog perspective. It has a continuous permafrost. It has a lot of these sulfur-rich, high saline springs. And despite the average air temperature being about -15 degrees, these springs are still active. So it's kind of like a polar desert environment. Now, from an analog perspective, we have been using this site as an analog for the Neokion-Hesperian transition on Mars. This is where it's kind of thought that there was sulfurous and saline water existing supposed to exist on the surface of Mars. And if we think about the process of that, back to that video I showed you earlier, during the Neokian period, there was liquid water on the surface of Mars, and many locations had rock formation which were rich in sulfur species. And then at the end of the Neokian, into the beginning of the Hesperian, the presence of the water declined, causing really saline-rich brines to form. And there's evidence of this where we look at the jarosite deposits in Gel Crater. So based on a model, so the SURA model, which is one that you can use to kind of predict analog sites and determine them, the Canadian High Arctic, the Axel Hyberg that we were looking at, was an ideal analog for the work that we wanted to do, which was very much looking at microbial processes within this sulfurous environment. So the first thing that we did was we wanted to know, how was it really going to be a good analog for what we wanted to do? So the first thing we did was we measured the chemistry, the fluid chemistry of the water at the site, and we plotted that against other known fluid chemistry. So Suzanne Schwenzer is a colleague of mine that we work very closely with. She had done a lot of work where she had modelled potential water compositions at Gale Crater based on MSL data, and also using Martian meteorite compositions. So when we plotted our analog sites, some of this work that she'd done, we found that they kind of plotted really closely together. We were specifically interested in the Lost Hammer environment, and this was very much home to a very much a saline, really sulfide and sulfate rich spring. as you can see here on the right, this is what Colour Peak looks like, and there's a variety of different springs which discharge into this deep gully. So the pH was very similar. It was written in neutral, about 7.3 to 7.9. So it kind of, we identified this when we compared it to the thermodynamical modelling of the fluid, of the evaporitic fluids. For Mars, we kind of decided this was a good analogue for what we wanted to do. So as a microbiologist, I was really interested in looking actually, well, okay, we've got this environment that has a very much similar, probably geochemistry to what early Mars could have been. So what microbial processes would exist? We know microbes would live in these environments. There'd been a few studies done previously. So I was interested in looking at what the active community was doing and what metabolism, so what energy it was using for energy within these systems. So what we know and what we did was we carried out some analysis using 16S analysis. So we used it looking at DNA and RNA, so looking at the active and kind of total community within these environments. And very much as predicted, you know, this is a sulfur-rich environment, and what we were predicted was that primary production in these environments would be very much driven by the high levels of sulfur species. So there's a lot of sulfur oxidation going on within these environments, and the diversity of the microbes reflected in that, with a lot of organisms associated with sulfur-related metabolisms. And when we think about this on Earth, you know, oxidation, when we think about sulfur oxidation on Earth, is normally either coupled to oxygen, so an aerobic metabolism, or to nitrate. So what we did was wanted to know actually how feasible this actually would be on Mars, because obviously an analog site's great, but it's not a true representative of what you would have within this Martian environment. So what we did was we did some calculations to determine the feasibility of this. So we used some of the modelled Martian fluid that Susanna Schwenzer had done, based on the evaporitic ones, based on Gale Crater. And what we did was we determined how much energy would be produced from sulfide oxidation within these environments. So we modelled it with oxygen and we modelled it with nitrate. So the rationale with that was, I know everyone thinks Mars is, you know, is a very anaerobic environment, which is true to a certain extent, but there are have been examples where oxygen has been detected in the Martian atmosphere. So we modelled that that amount of oxygen within these models and demonstrated that even with that low level of oxygen, this metabolism could be feasible. We also modelled it with nitrate. Curiosity has detected nitrate in ancient mudstones at Gale Crater. So there's a variation of composition between 70 and 1,000 ppm. And when we modelled it with both of that, again, the metabolism, you know, we demonstrated that this sulfur oxidation metabolism would be feasible within these environments. So this was great. So it kind of, rather than just looking at this as an analog site, we were able then to take the information we'd learned and use the information that we'd modelled to look at, to see how this was actually feasible. So applying it to that environment. Now, you know, these, I think, as I mentioned, you know, these Martian analog sites are great for kind of getting a snapshot of what's going on, but the parameters are not exactly what you'd have in these extraterrestrial environments that we're trying to study. So what we do is we marry them up with controlled lab experiments. So this allows us to control and contrast parameters, so we can look at how changes in temperature or pH can influence the habitability of an environment. The advantages of using lab simulations as well is that the samples are heterogeneous. So rather than having a natural variation, which can be problematic with some of these studies in analog sites, we can make sure everything is heterogeneous. And also, which is quite useful, particularly when you're looking at biosignatures, is that we can run abiotic controls as well in parallel, which again is something you don't think about field sites, but having an abiotic and biotic control just makes it really so much easier when you're trying to say that something is biotically produced. So Initially, when you look at the literature over the last 20 to 30 years, routinely analog lab simulations have been very basic and involve batched experiments, such as that on the left, as you can see here, which normally involves growing a microbe or a microbial community in a minimal medium, which is very much been developed already to support microbial growth, and also basalt. So basalt, although the Martian environment is pretty basaltic, a lot of these experiments just use terrestrial basalt. So there's a number of limitations for these experiments. Firstly, they're closed systems. We've talked about these large fluvial systems on Mars, so they're not readily representing those environments. The growth medium, again, is very much defined on what we know microbials will live. It doesn't take in the chemical conditions which we know that we can model to exist on Mars. And also, yeah, basalt's okay as an analog, but it's limited in how you can compare that to what is on Mars. So what we needed to do, so we've kind of really developed this technology. So to develop an analog site, a lab simulation experiment, it's really important to make sure that you define all the parameters that you put into these experiments. And a key part of this is the water to rock ratio interaction. So, as water on Mars would circulate, there's water in the Occian period through the regolith, the rock dissolves, liberating elements into the water, and these elements could be bioessential elements, it could be a energy source, so it could change redox states of chemicals. So understanding that water to rock interaction is really important. And from that, we need to pick a microbial community, which again is potentially could live within those environments based on analog work. And then we also need to make sure that the environmental conditions which the experiment is carried out really can simulate what we expect. So the first step with an experiment like this is picking your regolith, your Martian rock, as it was. So the regolith, the simulant, very much mimics the mineralogy of what's been predicted from from missions to the surface of Mars. So it's very specific on the location where it is. So for us, there's a lot of general regolith, Mars regoliths out there, so it's JS1, but we wanted to kind of home in on specific environments. So we've actually developed 4 different regoliths which are dependent on the environment and the site that we want to look at. So, for example, we have our global one, RockNest, which we use when we want to compare to kind of other studies which have used Martian simulant. We have a Passaroblus one, which is very much sulfate rich, We have hematite slope, which is very much based on the really rich hematite slope environment. And then we have a Shergotite, which we've used as kind of like our early analog simulant. So it's very much based on the meteoritic composition, which is one of the earliest meteorites that have been found. So we have kind of made these ourselves, so by just adding bits of different minerals together. And what is different than ours to the other regoliths that you can get is, firstly, as a microbiologist, I really wanted to make sure that we had the iron Fe2 to Fe3 ratios right, because that is really important when we look at microbes being able to use inorganic chemicals as a way of producing energy. And our simulants are really similar to what we predicted in the mineralogy. So all of them are less than 5% different total composition. So once we got the chemistry, what we also then did was that we wanted to develop the brine. So this is the water composition of the environment, as you can say, rather than using a defined minimal media. And for this, we used a thermochemical modeling called ChimXPT. And this is very much already used in kind of for understanding and studying brines and basaltic weather, which has been applied to some of these compositions on Mars. So it's been used, so Susanna's used it quite a lot to model the potential water composition of Gale craters. So what we've done is we initially inputted pure water, we dissolved the rock based on the rocks that we showed you before, the simulant, and then we just started to develop this simulant. And in this graph here, you can see the basic composition of the brine water that we used. There was no nitrate in our brine based on the chemistry that we added into the system. So what we did was, based on the Stern paper, was that we added nitrate to the medium, and for that we used a 0.1% weight percent. And then finally, the last part of the puzzle, I suppose, for the experiment was the microbes. I think it's quite interesting to think when you're looking at this kind of transition, you know, the environment that we're looking at on early Mars, sometimes you don't actually have to go to some of these really extreme environments. So what we wanted to do, what we do for these analog work, for lab work, is we use microbes from the intertidal zone. So basically we're looking for a microbial community that can survive in low temperatures, you know, relatively, you know, at 2% saline, a variety of redox states, can support evaporation. So for us, the intertidal zone, it's quite close, it's easy to get to, is what we have been using for these experiments. And from a community perspective, it's actually ideal because it has a whole selection of microbes using different metabolisms. So you have your chemolithotrophs, so microbes again that utilize inorganics. You've got microbes that can use organics as well. So for us, we've been using these because it's a huge mixture of microbes. So there's not a big selection that happens before we do this. So the first step in this experiment is that we take the sample and we grow it up in the brine. So we start with really concentrated brine. So we take a huge amount of simulant and we dissolve it in a small amount of water, so we can starting to develop the microbial community, getting used to this kind of Martian brine environment. And then after subsequent subs into different bottles, we then dilute it. So we decrease the amount of concentration of the brine. And on the right, you can see here, as this actually impacts the microbial cell numbers. So you can see a drop off after about stage 4, and this is when the amount of rich microbes, the rich media kind of decreases. So it's not so much mineral available for the microbes to utilize. And what impact does this have? And I think this is something which is really important, because there's a lot of papers out there in the literature at the moment which they just go straight from a analog site straight into a Martian simulant experiment and talk about how microbes can grow, etc. But actually, if you look at the diversity at each stage of this process that we've done, this is actually over 7 months of subbing the cultures every month, is that you actually see that the community develops and evolves over this time. So, for example, if you can see here, when we look at the end note community, it's very much dominated by sulfur-reducing microbes and acetogenic microbes, which is very different to what we originally inoculated it with in the beginning. So I think when we look at in the literature, when you read about analog, you read about some of these lab work, it's just very important to look at what microbes they actually put into the system. Okay, so I think the final step of the experiment is, as I mentioned before, is about the environment, the environment in which they live. So rather than using those little bottles, we've developed this flow-through system, which allows us to look at kind of larger water-to-rock ratios. So in the reservoir, we put the brine, which we've modelled. In the reactor, we have the rock simulant and the analog community. And what we can do is pass the liquid, the brine water over the rock, so very much simulating a flow-through system. And what we can do is that the way that we built this is that it's in a high pressure vessel. So this means that we can look at changing different parameters, looking at different depths on Mars. So for example, we can simulate point right up to 10 kilometers under the surface of Mars, where the pressure and temperature as well. So it's kind of a novel bit of equipment that we've developed here, but it's really useful when we start wanting to change from looking at ambient conditions on Earth. Very much we can look at these subsurface environments where potentially life does exist on Mars. And this is really useful. So when we can grow these cells then continuously for a long period of time, so we can see that there's growth. Cells still manage to grow within these systems. What we find is that the pH, because the microbes grow, they can produce acidic metabolites, and this decreases the pH. And although some of the regolith can buffer some of that change in pH, that does still influence the pH of the fluid slightly as well. So you can see variations in the pH there as well during the abiotic and biotic experiment. And again, the diversity is something that changes. So when we compare the inoculum to the flow through into the batch, so the batch is the closed system, the traditional way of doing it, again, you see changes, differences between the microbial community, which is something when we think about looking at this, is really important when we think about habitability, is to look at it in the context of the environment. So if we're looking at fluvial systems or very much small closed systems on Mars, that does impact the kind of microbial community and the chemistry which you will have. And I think it's really important when we think about this to think about it as a site-specific way of looking at. And finally, I think the major point about using these kind of lab experiments is it's really useful to look at biosignatures. So biosignatures are molecules that are produced uniquely due to biological activity. So this can be anything from secondary minerals that are produced due to changes in the pH caused by microbial growth. It could be caused by organics, the microbial cells themselves, changes in the isotopes, for example. And I'm just going to touch on here some of the changes that we showed. We're able to look at the surface of the minerals from those experiments. So even here, when you look at the abiotic and biotic differences between the experiments, you can see that there's definitely differences between some of these. And that could be due to the cells being used as inoculation points, the cells causing changing acidic, causing secondary alteration minerals to produce. There could be a whole host of this. But I just wanted to show the take-home message here, how the biotic and biotic systems change. But probably, again, which is really important to note, is also the closed systems with the bottle experiments are very much different to the flow-through systems as well. Again, it's really important to understand when we think about habitability, looking at the environmental context in which they are within Mars, because that influences not just the habitability in the microorganisms, but also the potential biosignatures that they can produce. So I think my final take-home message is, when we're looking forward, this diagram here on the right about the missions over the last, the missions we've had over the last 10 years and looking to the future, how we need to look at Earth to start to understand some of those processes, and then applying those to the lab, where we can start to really home in on how life could exist within these extraterrestrial environments and what influence that would have on the potential geochemistry and the biosignatures, so evidence of life. And I think this is all really important when we look forward to these missions, and particularly with Mars sample return, that we'll be able to really home in on some of these changes and hopefully be able to identify evidence of life on Mars. Thank you very much.